Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with Shatin Sawati, and I'm here to discuss about the Mendel's laws of inheritance today. This is my second lecture from in my inheritance series. I have given you a complete and detailed concept of dominancy, recessiveness, phenotype, genotype. So must watch my first lecture. This will clear your concept related to the genetic inheritance. So today I'm going to discuss the most important thing, that is the monohybrid cross and dihybrid cross, as well as I will discuss in detail about the law of segregation. So first of all, what is monohybrid and dihybrid? But before starting my lecture, I have a very humble request to all of you to do subscribe my channel if you did not, and like my videos and share my videos. You're sharing my videos, you're sharing the knowledge, so keep watching, keep sharing knowledge, and kindly do viral the education content, because we need education to grow and survive. We need education to develop. Okay, so first of all, what is the monohybrid and the dihybrid? Monohybrid, mono mean one and the di mean the two. So what does it mean that monohybrid cross and the dihybrid cross? So first, monohybrid. You can see here, this is the monohybrid cross. The cross involving one contrasting character or trait. Is it clear? So we are considering just one character, like the seed color. Seed color. This is true breeding yellow cross with the true breeding green. Is it clear? So just one character trait is involved in this cross. So this cross is the monohybrid cross. Is it clear? Yellow cross with green, and the hybrid in offspring is what will be the offspring? That will be yellow, because the yellow color is dominant. And I have discussed in detail about the dominancy and recessiveness, homozygous, heterozygous. So must watch my first lecture related to the basic concept of the genetic inheritance. In the second figure, you can see here the dihybrid cross. The cross involving two contrasting character, mean two traits at a time. Like we are considering here shape of the seed as well as the color of the seed. Is it clear? The shape is rounded and the shape is wrinkled, and the second character color, yellow color cross with green. So two contrasting characters are traits involved in this cross, and the cross is known as the dihybrid cross. So the concept clear now. Monohybrid cross involving just one or single trait or character at a time. The cross involving two characters, like the seed shape, here considering, and the seed color. But in monohybrid, just one trait. You can take just one, like the seed of the shape or the color, just one character. So monohybrid cross involving one, and the dihybrid involving two. The concept clear now? Okay. So you can see here the other example of the monohybrid, and also the definition is available here, and you can make your own notes. So hold down notebook, pen and pencil, and note down all the lecture. Okay. Here the shape of the color of the, you can see here the color of the pea plant, green and the yellow. The green color is dominant over the yellow color. So this is just one character or trait involved, that the cross is known as monohybrid cross, which I'm going to discuss in detail in law of segregation. Law of segregation, I'm going to share here. The reason is that this law involves or follow the monohybrid crosses. Is it clear? Law of segregation follow the monohybrid. So that's why I'm sharing with you here. The law of segregation states that alleles segregate randomly into gametes. Segregates means separate from each other. Is it clear? So that alleles, you can see here the two pairs, one pair for the green color and the other pair of allele for the yellow color. Is it clear? Two genes, one gene for green color and the other gene for the yellow color. But these two genes have how many alleles? 4 alleles. Is it clear? So GG, this is the dominant and this small g, this is the recessive. So the law of segregation states that alleles segregate, means separate, randomly into gametes. You can see here, when the gametes produce, the two genes segregate separate from each other in each gamete. Two gametes are formed, this one, and the alleles segregate are separate randomly. When gametes are formed, each allele of one parent segregate randomly into the gametes, such that the half of the parent's gametes carry each allele. Is it clear? So this is the diploid condition, and this is the haploid condition. Gametes are haploid. From the parents, when gametes produce, these gametes are with just half alleles. Is it clear? So this is the law of segregation that states that the alleles segregate randomly into gametes. The concept is clear now? Okay. Now, in the image, the single trait being observed is pot color. As I have shown you, we are here considering the just one character, that is the pot color. As I have discussed that law of segregation follow the monohybrid cross. So we are here considering the example for the pot color. As you can see, the green pure breeding green pot color cross with the pure breeding yellow pot color. Is it clear? So this cross involves just one contrasting character, and this is the example of the monohybrid cross, and I'm here discussing in the law of segregation. Is it clear? Okay. So the single trait being observed in the pot color, the organism in this monohybrid cross are true breeding for pot color. True breeding organisms have homozygous allele for specific trait, must keep it in your mind. In this cross, the alleles for the green pot color is completely dominant over the recessive allele for the yellow pot color. Here the dominant trait is the green pot color, and the yellow is the recessive. The genotype for the green pot color, as you can see here, the capital G, capital G, and for the yellow color, this is small g. The cross-pollination between the true breeding homozygous dominant green pot plant and the true breeding homozygous recessive. As you can see here, the true breeding green cross with the true breeding yellow. Is it clear now? So, what will happen? The offspring with phenotypes of the green pot color. So the offspring in the F1 generation, you can see this is the first parental generation, and after gamete formation, what will happen? These gametes fertilize with each other and produces the capital G small g, and this capital G small g phenotype will be green pot color, and you can say that the genotype will be capital G small g. So this is the heterozygous. Is it clear? All genotypes are GG, capital G small g. The offspring of our F1 generations are all green because the dominant green pot color is the recessive. Hide the recessive yellow pot color in the heterozygous genotype. Is it clear? Okay. Now you can see here, this is the G means the true breeding green when crossed with the true breeding yellow. And all the offspring in the F1 generation will be green in color, phenotypically green, but genotypically they are heterozygous. Must keep it in your mind. In the F1 generation, all the organisms, all the offspring are what? They are green phenotypically, but genotypically they are different from their parents because they are heterozygous. Is it clear? As the parents are what? Homozygous. Parents are homozygous and the offspring in the F1 generation are heterozygous. Is it clear? Okay. Now F2 generation. Should the F1 generation be allowed to self-pollinate it? Now, how it will possible? I will show you. The potential allele combination will be different in the next generation. I am showing you here. You can see here. Now, F1 generation, that is the heterozygous What heterozygous green? Heterozygous green pot plant F1 is self-crossed with the other F1 plant. So this is the self-cross between the self-cross of the F1 generation. This is what? This is the self-cross of the F1 generation and the second parental. This is considered as the second. That first one was the first parental generation that produces the F1 generation, and now this is the second parental generation. Now second parental generation, how it will be possible? By crossing the F1 with F1, means the F1 is self-crossed. Now what will happen? Here you can see how many gametes will be produced. Capital G, yes. Small g, this one. Is it clear? The other parent, capital G and small g. So you can see here the gametes formed this from this one. Is it clear? Now what will happen? Now the F1 is self-crossed with F1. You can see when you cross the capital G gamete with the capital G gamete of the other parent, then what will happen? The homozygous GG, capital G, capital G. So this is the this is what true breeding green, just like the parent, first parental generation. Now the second one, capital G, small g, they are like the second parental generation, which are heterozygous green. Now this is also the heterozygous green, and this is the, sorry, this is mistakenly written as the capital G. Here is small g. Here is small g, small g, small g. Is it clear? This will be here. Is it clear? So small means recessive. So this is also like the first parental generation, means they are homozygous recessive. Is it clear? So you can see here how many homozygous green, how many heterozygous green, and how many homozygous recessive. So you can see just one homozygous green, two heterozygous green, and one homozygous recessive. So the one homozygous, if we say that the homozygous dominant, it means this is the genotype. If we say that this is green, this is green, this is green, so this is what the phenotype. So phenotypic ratio and the genotype. Phenotypic ratio, how 1, 2, 3 green. 3 green, so phenotypic ratio is the 3 and 1 yellow, and this is what? 1. 3 ratio 1. 1 yellow. Phenotypically, when you observe, what will happen? 3 green, 1. But genotypically, how many? 1 homozygous dominant, 2 heterozygous green, 1 homozygous green, 2 heterozygous green, and 1 homozygous recessive. So 1 ratio 2 ratio 1, and the 3 ratio 1 phenotype, genotype 1 ratio 2 ratio 1. Is it clear? Okay, very interesting. Now you can see here the second example. You can also solve, this is also for the yellow seed color and the green seed color, just like producing the gametes from the first parent, then the F1, sorry, so F1 is self-crossed to get the F2, then the F2 will be like that. So I hope so the lecture will be clear upon you people. If you have any question, kindly write in the comment box. I will be back, and thank you. Till then, Allah Hafiz.